few more people trickling in, but we are recording this meeting, so people who come in late, or maybe if some universities are on spring break, those people will be able to watch this video later. So today we're going to listen to a webinar presented by Dr. Paul Gepts. He's a member of plant science at UC Davis, and his major interest is in the genetic improvement of phaseolus beans. And he has been primarily involved with activities developing the genetic and genomics information necessary to support breeding programs. His research uses an evolutionary approach to help understand how genetic diversity is organized within the genus, with a particular emphasis on the common bean. So specific research aspects he's involved with include the development of bean genetic and genomics tools, identifying domestication and geographic patterns of genetic diversity in beans, and documenting the loss of genetic diversity following domestication. He has also conducted a lot of research looking at coevolution between beans and their pathogens, and looking at gene flow between wild and domesticated beans. In addition to this involvement with the bean breeding program at UC Davis, he also somehow manages to find time to conduct and disseminate research and also to teach. He's also the main editor of a book published this past year titled Biodiversity in Agriculture. And his talk today is Insight to Farmer Management of Genetic Diversity as a complement to ex situ conservation. So with that, I'll let Dr. Gepts get started.
Well, thank you very much. Can you hear me? If you don't, I can try to speak louder, but thanks very much, Sarah, for the introduction, and it's a pleasure here. I have to apologize about the miscalculation on my part last fall. And this time, I think I have it straight, actually. The topic I'm going to talk about today is really fairly recent. It has been developed over last years, and in part it came out of a project that was funded by the McKnight Foundation in Mexico, with a whole group of people working not only on beans but also on corn and on squash. And the focus in beans was really on gene flow. And out of that came a realization that we need to take a stronger look at how farmers actually manage their seed stocks and what effect that may be. And conversely, can we detect the effect of farmers at the genetic level on the genetic diversity of their seed stocks? And we have used then specifically molecular markers to address this. And so what you'll see here is a combination really of molecular studies, but also studies in the field that rely on samples provided by farmers. Okay. So I'm going to the next slide. So what I will do in the introduction, I'll raise that question specifically about is there a role, if any, of farmers in maintaining and shaping crop genetic diversity? We'll then provide population genetic evidence for farmer role, also molecular evidence from a specific gene for domestication, and then I'll end with a discussion on looking at what the consequences are of our findings. So my view about, so a lot of this comes about from studies of domestication, which has been kind of the major basic research interest. And you can define domestication as the process that has a selection process that has taken place as a consequence of the cultivation of wild plants. And the cultivation in itself is a very different environment from the wild environment, and as a consequence there have been major genetic changes. And the genetic changes actually, the extent of the genetic changes depend on factors that are related to plant or animal themselves. I'm seeing here a few, such as morphology, behavior specifically for animals, but also genetics, physiology, the capacity to respond to evolution, and so on. The environment, we know that agriculture resulted as a consequence of a major event in climate change, which was the end of the last ice age. We see, for example, that the importance and contrast between dry and humid seasons may be important. The diversity of niches and so on also plays a role. And then what makes this crop evolution and domestication studies very interesting is, of course, the human involvement. But as plant scientists, we are focused primarily on the plants themselves, somewhat on the environment and somewhat on humans, but actually fairly little when you look at the literature. And so the study that I will be talking about today fall into that left bottom triangle here on the left side to see what is the role. Now it's fairly difficult to look at the role of humans 10,000 years ago, and that's the realm of the archaeobotanists. But I think that we can look actually at the role of humans contemporarily, and then try to make some inferences as to what the selection process might have been several millennia ago. But also it has an importance today to look at the genetic diversity of the seed stocks managed by farmers, and in terms of in situ conservation. Okay. So the next slide then shows, actually, in the case of beans in Mexico, and this is a photo of a colleague, Daniel Sisumbo from Mexico, and what you see is that the farmers actually pay a lot of attention to the seeds. Usually, even though they harvest what is a mixture, which you see there on the right bottom, they eventually separate out the different colors and different seed types, and they usually sell a pure, and I say pure in between quotation marks, on the market, and it fetches a higher price than just a mixture. And for many of these farmers, that is the extent of the selection. Now you might say, well, do they do selection in the field? Well, as far as we can tell, in most cases they don't. They certainly make observations, but they don't. They harvest in bulk, and so they don't do selections in the field. And so that's what I alluded to, is that do we know how do we know that there is an effect of the farmers themselves, actually? And that this is not just a kind of a bulk harvesting with limited, where there may be natural selection, but there limited human selection. Okay. So the fact that people select doesn't mean that actually selection is going to be effective, and that's true for plant breeders, but also for farmers. So it also has an implication in terms of ex situ versus in situ conservation. You're familiar with the different gene banks. We have the CG system gene banks. In the US, you have the USDA as the National Plant Germplasm System, and you see on the right side at the bottom here some of these stations of the USDA. In different environments, depending on the adaptation of the different crops and so on. And there are also national, international banks. Brazil, for example, has a whole series of gene banks as well. In Europe, you have them, in Japan, and so on. Okay. But what we are interested in is primarily, so this is the case of the Brazilian system, and you see that it's a fairly large size system as well. So the alternative to this has been one of the problems with these large collections is that it's very difficult to enter the collection. And where do you go, actually? So say that you're interested in, I would like to look at drought tolerance, for example. So which of the thousands of accession might be useful? And that would call for screening, but it's probably way too much. If you have 10,000, 20,000, even to 100,000 accessions, how do you identify the specific accession? So people have suggested this core collection concept, which is about 5 or 10% of the general collection. Okay. And so the assumption there of this core collection is that somehow in a fraction of that core collection, we are going to find some materials that may be of interest, for example, that may be drought tolerant, and it would be easier to screen for drought tolerance this smaller collection. Part of the question, especially if you deal with domesticated materials or cultivated material, is you assume that there has been some adaptation to local conditions that are drought related. For example, reduced precipitation, higher evapotranspiration, and so on. And that if they're grown by the farmers, it is because they are adapted to these drought stress conditions, for example. So that is something that we need to check whether there is actually effective selection by local farmers, okay, to their particular conditions. So what does the data then tell us about the effectiveness of selection? And in the case of beans, primarily from our data in Mexico, actually. So I have to tell you a little bit about beans. I'll keep it to a minimum. I can talk for hours about beans. But Phaseolus vulgaris in itself is the major species. It's the most important one economically. It's widespread all over the world. And basically, it resulted from two separate domestications. One in what we call Mesoamerica, and we have identified recently also what is presumed center of domestication in west central Mexico and what is now mainly the state of Jalisco. And then the other domestication took place somewhere in the southern Andes, anywhere between southern Peru, Bolivia, and Argentina. And so you have southern Peru here, you have Bolivia, and then you have Argentina. And so all the bean varieties, or the common bean varieties, come from one or the other domestication. So if you go to a market today, you will see at the right, you see, for example, navy beans, black beans, pintos, small reds, or pinks. They all come from the Mesoamerican Domestication Center. On the other hand, if you would buy kidney beans, yellow beans, or even these so-called cranberry beans, and the majority, but not all of them, the majority of the green beans, they originated from the Andean Domestication Center. Okay, and then there is this yellow area here. On one hand, there is data that show that they contain the ancestral genes for the major seed storage protein called phaseolin, but the population genetic data shows a somewhat different picture. It might be just a migration and dissemination from the main wild populations, which are located here in Mexico. So it doesn't really matter for our discussion here, but the status is of the yellow group. Remember that there is two major domestication, Mesoamerican and Andean, and all these are derived from one or the other domestication. So a first set of studies then was conducted in the central part of Mexico. You see here in the left upper corner, the central part of Mexico, and the circles actually are different sites in that region, and they differ by the distance between the domesticated field and the wild population. And so it goes between 10 meters up to 110 meters. And the question was, do we see, for example, evidence of gene flow between wild and domesticated, and especially in the wild populations? Now, in order to determine that, it is somewhat difficult, and one of the difficulties is that you have a common ancestry. Okay? So both wild and domestic have a common ancestor, so you see also shared alleles can be due not only to gene flow, but just to common ancestry as well. So in order to separate the confounding effect here from common ancestry and gene flow, we took this distance aspect here. So what you would expect is that at the site that is farthest from the domesticated field, you would see much less evidence of gene flow than at the sites that are much closer, 10 or 15 meters, for example. So you can look at genetic diversity in the wild population and genetic diversity in the comparable domesticated type population. And what the results show, so you see here a number of variables, seed weight, for example, and seed weight coefficient of variation. Why do we do that? Well, because wild and domesticated types are distinguished by their seed weight. Domesticated types have much larger seeds. The wild types have much smaller seeds. And what you see is that the seed weight actually, as the distance increases, and the abscissa here, the x-axis is the distance from the field, you see that on average, there is a decrease in seed weight. The seed weight CV actually would be a reflection of the segregation you would expect if there is gene flow. And so what we see here also is that the CVs tend to be higher near the field than away from the field. Then there's a number of molecular parameters like the percentage of polymorphic loci also is higher because presumably of crosses between wild and domesticated type, and then it decreases as we move away from the field. And the same thing for these DNA diversity and so on. So what we see is that definitely that there is this aspect of gene flow from the field to wild types, and in part this is managed actually by the farmer who tolerate the presence of these wild populations in the vicinity, or even sometimes within the field in Mexico. So the next slide here is also, this is a different study, and where we actually measured the migration coefficients in two directions, from the wild to domesticated and from the domesticated to the wild. And what you see is that the levels of gene flow are not that high. I mean, they're below 1, but that the level of gene flow from domesticated to wild is about 3 times higher than in the other direction. And this is something that we have seen repeatedly, and also has been seen in lima beans, for example, which also grows in Mexico, and as well as in squash, actually. So it is an interesting observation that, yes, there is gene flow, but it is predominantly from the domesticated to the wild. And there are a number of potential reasons for that. One reason is that the domesticated populations are usually much larger than the wild populations. So there is a pollen mass that is much larger. The wild traits are usually dominant. So in the cultivated field, there's a potential with the farmers actually for a strong selection against wild alleles, much stronger because of the visual observation, but also because of the dominance of the wild traits. So these are some of the two major reasons. But again, here there is opportunity for the farmers to actually select or tolerate, select against the wild type or tolerate them. And what you see here, this photo, is actually quite interesting. You see the large pods here. These are domesticated bean. And here immediately to the left, the smaller pod is actually a wild bean pod. And so the question arose, why do they remain distinct in spite of the fact that they physically grow together? And one could say, well, common bean is a predominantly selfing species, and therefore you have little gene flow. And what we see is that there is gene flow, but there is probably selection after the hybridization events for maintaining either the wild type or the domesticated type. Okay. This gives you an idea. For example, I've talked about, the farmers grow beans and they can even tolerate wild beans. What you see here is again a photo of Daniel Sisumbo, and he the two photos here to the left are the mixtures of two different farmers. So this is Benjamin Rico to the left, Roberto Sisumbo to the right, and you see that they typically plant mixtures of different colors. One to the right, actually, all these beans that are called frijol coyote here at the bottom are actually, the farmer considers them to be wild beans. Okay, so you have this gene flow, but also you have the management by the farmer who influences the level of gene flow and how much actually gets retained as a consequence of gene flow. Here is another example you see here, this is the on the right side, the right panel here, you see frijol coyote. That means these are all various forms of wild beans that are tolerated, and you see that the table here actually shows the relative sizes of wild, weedy, and domesticated average seed weight here. And they also actually introduce this Anita is a commercial. It's a public variety, but improved variety of the preferred type on the market. So these fields of beans in Mexico are very often a mixture of wild beans, land races, the original varieties, and then improved varieties. And it is really the role of the farmer actually here. Some of them maintain very actively these mixtures of seeds. Interestingly, then, is that there is actually a fair amount of diversity. You can look at the wild types, the land races, and then breeding lines or improved varieties, and you look at overall diversity, and you see the large age here, the values are similar between wild and land race types. So normally in domestication, what you should consider is that there is usually a reduction in diversity, so there's much less diversity in the domesticated gene pool compared to the wild gene pool. Here we don't see that. So the farmer actually, through the management, is able to maintain a fairly high level of genetic diversity. And what has changed, however, is the distribution of this genetic diversity. In the wild type, it's mostly between populations, whereas in land races, it's within the population. That's what you see here with the age values and the GST values. And you also see that here, the N value from the migration, the number of migrants actually, is higher in the land races compared to the wild types. And so the gene flow here, you have to remember that the gene flow occurs not only by pollen, but also by seed. And what you see probably here, this higher value for land races, 1.40, is due to the exchange, the introduction and exchange of materials by farmers in between fields and with neighbors and so on. Okay. On the right here, you see kind of different situations. Here you have the top here, you have a wild type grown along a fence, and you have the field with domesticated beans and corn and squash growing inside the field. And then here at the bottom, it's more a situation where you have a larger distance between wild types and the field. The field is actually in the bottom behind the tree you see in the back there. Another way to look at these data is to do a spatial autocorrelation within Mexico. So you can start, for example, with at the southern Mexico, Chiapas, for example, and then you look at other sites in Mexico where you have wild and domesticated types growing together. And the two top are wild versus weedy, and the bottom is domesticated. And you see that these autocorrelation coefficients are actually much stronger at shorter distance, which kind of highlights again the uniqueness and the isolation of individual wild populations, whereas in the domesticated types, it's a much more even distribution of genetic diversity. So clearly, there has been an effect of domestication on the distribution, the geographical distribution of genetic diversity, but also, even today, the farmers, through their active management of seeds, whether they are wild, land races or improved types actually affect the distribution of genetic diversity. So another study in the same region, actually in Mexico, by Sarah Barber, who was a former student, master's student. She then started inquiring with the farmers. And interestingly enough, is that she found that there are two series of land races. They're recognized by the farmer. You have early varieties and late varieties. In the left column, you see a list of the characteristics of some of these late varieties. So it's a late harvest in November, December. The planting is about June, July. Okay, that's when the rains start. They have thicker seed coats, so that they are longer, takes longer to cook, but they also have a longer storage time. And then you see the list of these are names of different land races, different seed colors mainly. And on the right, in the right column, you see the early harvest, thinner seed coats and shorter to cook, but also shorter storage time. And so one of the major determinants that farmers look at is really the earliness. And we see that elsewhere also, and for other crops as well, is that earliness is almost more important than yield per se. In that, I think, and this is probably related to food security and the need to have an early harvest to replenish the grain stocks. Okay. But again, it's a way that farmers actually are able to manipulate the genetic diversity. Then the most recent study in this regard was conducted by Margaret Worthington, who is another master student, and colleague, Dr. Daniela Soleri of UC Santa Barbara. And Margaret has now gone on to North Carolina State, and she is in the plant breeding program. And what they did was to look at a specific village in the state of Oaxaca, and you see here, this is a Google Earth representation, and the circles indicate different fields, and the labels then indicate farmer, and then you see 1, 2, 3, and then some farmers are represented 2 times. And you have a first group of fields here near the village itself, and then you have another another group here that is actually, it's difficult to see in the picture here because there's a lack of perspective, but it's actually at a lower altitude, and it's closer to the river, which you see here flowing in the landscape. So here there was an opportunity to look at genetic diversity at different altitudes. When you look at the materials themselves, for those who are familiar with beans, actually there were 3 groups, taxonomically, morphologically. You have the green group, which is actually a different species. It's 2 different species, Phaseolus coccinea, the runner bean, and Phaseolus dumosus, which is the year bean, both of which are domesticated also, but much less widespread. And then you have a group, the blue group here represents a ecogeographic race. And you see they are uniformly small-seeded and black. And this is what is called race Mesoamerica, and it's representative of lower altitudes, warmer and wetter. And then you have the red group, which is the traditional ecogeographic race grown in the village, and that is race Jalisco. And you see that there is variation. The seeds are generally larger, also variation in seed color as well. And so the question arose, well, if you see these 3 groups, where are they grown in that village. Now, if the farmer didn't have a role in picking where they would grow and which mixtures and so on, you could say, well, we're going to find them more or less at random among these 10 fields that they looked at. And their composition would vary from one field to the other without rhyme or reason in terms of adaptation, altitude, and so on. So how can we find out whether that is the case? Well, you can look at it morphologically, but also you can look at it molecularly. And so Margaret actually ran a fairly small number of microsatellites, about 10, and was able to distinguish actually many of the materials this way. And she ran what is called a structure analysis. You're probably certainly familiar with that. And these are the results here of that structure analysis. And you can start with k equals 2, then 3, 4, and so on. And the result actually of k equals 2 is that you separate the red, which is Phaseolus vulgaris, from the green, which is Phaseolus coccineus and Phaseolus mimosus. Now, if you look at that structure graph, you'll see these white vertical lines. They separate different fields. And so there are 10 fields that were analyzed. And what you see is that the delimitation between Phaseolus vulgaris and Phaseolus coccineus is really, there was one field of Phaseolus coccineus. Okay. If we increase k from k equals 2 to k equals 3, what you then see is still the separation with Phaseolus coccineus and mimosus, but then you start seeing the subdivision within Phaseolus vulgaris. And the subdivision coincides with the limits actually of the field. So what it suggests is that farmers actually tend to plant according to what we recognize as taxonomic subdivisions, either the species or the ecogeographic races. And that clearly suggests then that they are aware of these broad morphological and ecological subdivisions. Okay. You can continue k equals 4 to even k equals set, and what we see, we usually do that even though the Avano test tells us that it's 3 or 5, but just to look at the strength of these groups identified by structure. For example, the fact that the coccineus and mimosus group, the green group here, is maintained all the way through k equals 7 suggests, yes, this is a really strong separation. But you have others, for example. You have the group here, the third column, the third field here. It maintains its identity all the way through k equals 6, and even we could say argue k equals 7. So some of these ecogeographic subdivisions, although they are subspecific, are represent a real biologically important subdivision. Okay. So in the next slide is actually the same or similar representation, but in this time, so each of the circles represent a different field, but the ordinate is the y-axis is the altitude. And you can clearly see that there is a separation. We have the group of lower fields. Then we have the kind of group of fields at mid-altitude, and then you have the third altitude, and that is Phaseolus coccineus mimosus. And overall, what we see is that the genetic composition of the fields actually matches what we know about the adaptation of the three taxa. Phaseolus coccineus and mimosus are definitely adapted to cooler climates. That's why you see coccineus, for example, grown in the US, for example, in cooler areas, but also in Western Europe and so on. And in Mexico, you typically find it also at high altitudes. In mid-altitude, we find race Jalisco, which is an ecogeographic race that is characteristic of the humid highlands. And then at the lower altitude, you see the race Mesoamerica coming up, and that is, like I told you, it is an ecogeographic race that is characteristic of the lower, warmer altitudes. Okay, so the farmers are aware not only of the morphology but also of the adaptation, and they tend to plant their field according to this morphology and this adaptation. One of the questions is actually what is going to happen if the temperature continues increasing. Will actually Coccinus still be able to survive, or will it have to move north, for example, to achieve a cooler temperature? The other question is what is going to be the reaction of farmers? Are they going to include the race Mesoamerica, which is adapted to higher temperatures? And what we see is that at this stage, probably what they did is not as a response to global climate change, but really to become involved in the market. Because race Mesoamerica, I showed you, has these small black seeds, which are very popular on the market in Mexico, at least in some regions. And so what they are doing is to plant these small black beans to participate, to be able to sell them on the market. But at the same time, you start seeing that there is gene flow between the two races in the farms. So you see one farmer here who already planted what is basically a race Mesoamerican material at mid-altitude. But also you start seeing gene flow as measured by chloroplast DNA. So in addition to the 10 nuclear SSRs, she also looked at chloroplast SSRs. And you can kind of tell here the numbers below the circles represent the haplotype of the race Jalisco. So this field here has a diversity of 0.30, H value 0.30, but has 0 chloroplast DNA of race Jalisco, as you would expect. But then this other field here, you see already 30% of the chloroplast DNA is not from race Mesoamerica, but it is already from race Jalisco. And so clearly there seems to be introgression going on. And the risk probably among some farmers, it suggests that some farmers actually are actively leading some type of introgression between the two ecogeographic races. So further evidence that it matters, the farmers do play a role in the genetic diversity of their seed stocks. So to conclude, to kind of summarize what I've presented to you, is that certainly farmers have an awareness of the adaptation of the different germplasm units in terms of climate, phenology, and morphology. What type of selection, that is not very clear. It suggests that at least they have a role in the maintenance of the type. They know how to distinguish between wild and domesticated types. The question is whether there is a directional selection. Do they try to improve yield, for example? That is not very clear, and the further research is needed for that. Certainly, they are able to maintain genetic diversity, and it's a dynamic system. It constantly changes. They are able to, and they're willing to incorporate newer varieties when they see a benefit to it. And we saw that for the Anita type, which is a Flordemalio type, and the small black types here. And that raises the possibility that there is actually a niche for intervention by participatory breeding, where we would take the abilities of the farmers and combine them with the ability of the researcher to generate segregating populations, for example. And we would need to look into what is the improved germplasm, what are the selection procedures, and so on. Now, I would like to turn then to, this is the kind of, you could call it population genetic data, but turn to, you could say, a more abstract or further removed from the field, molecular data, to show that farmers do select and that they do so in a repeated way, actually. And that these data come out of our attempt to identify domestication genes in beans. And for one of them, we actually have a very kind of solid picture. That is the so-called determinacy trait. So let me explain first, what is the determinacy? So the normal, you could say, bean type is indeterminate. And indeterminate means that they continue producing leaves and inflorescences until senescence. And basically, the growth pattern of plants is a modular one. And so a module in this case is an internode, a leaf, and then an inflorescence at the axil of the leaf. And then you have the next module when you have the internode, the leaf, and the inflorescence. And it continues like that until senescence. In a determinate type, you have a terminal module, actually. So you see you have internode, leaf, inflorescence, and then you have the terminal module, which has an internode, the leaf, and then a terminal inflorescence. And so the effect of that, and this is conditioned by a single gene, a recessive allele, is to actually shorten all the branches, induce earlier flowering, and also a shorter flowering period. So on one hand, it has a major effect on plant growth. So it has a basic interest. And then on the other hand, it also has definitely a practical importance because of the shorter flowering period. You see that very often green bean varieties are determinate because it makes it easier for harvest. All the pods have more or less the same age, which would not be the case in an indeterminate type. And so you see here determinate types in common bean, but also in lima bean. And you see the inflorescences here at the that stick out of the foliage. Okay. So if it's a single gene, actually, can we identify this particular gene? And look, and what is of interest to me is not so much the gene itself and how it acts, but its polymorphism, its diversity. In order to find this, what we did was to do a candidate gene search, and we took genes from Arabidopsis. There were 12 here, all involved into flowering time and so on. Of these 12 genes, we were able to identify amplification products for 8 of them. And I won't go into the details. It has been published. 7 of them were mapped. And actually, 3 of those 7 actually co-segregated with phenotypic traits. And what we were very happy about is that at least one of them co-segregated with the phenotype, the determinacy phenotype. And that was a homologue of the TFL1 gene, terminal flower 1 gene of Arabidopsis. In beans, there are actually 3 loci with sequences related. And the one that actually co-segregates with determinacy, the phenotype, is the PVTFL1Y gene. So that's what I'm going to focus for the next slides. So we were able to map it, and The phenotype is actually called fin, so finite, finis in Latin, finite in English, and you see PVTFL1Y here. But there are two other loci, one on linkage group 4 and the other on linkage group 7. And the one on 7, we think that it actually co-segregates with a locus that was identified by artificial mutagenesis and also produces determinacy. Further evidence that this PVTFL1Y is the actual locus underlying the phenotype came from a co-segregation in a large population, 1470 individuals. Also from quantitative PCR, where we see no detectable or very low expression, which is consistent with the recessive nature of determinacy. And then we did actual transformation of Arabidopsis, the mutant or TFL1 mutant, with a functional gene from beans, and we're able to restore the wild-type phenotype in Arabidopsis. Okay, so clearly, I think all of this strongly suggests that this is the gene. Then we started looking at the diversity of this the phenotype in relation to the genetic background. And what you see here is a neighbor joining tree. It's split in half. You see the left and the right. The left is the Andean group. So you have wild forms from the growing in the Andes, and then the three ecogeographic races here, Andean ecogeographic races. And then you have wild Mexican types, or wild Colombian and Mesoamerican types, and then here the three ecogeographic races, races Jalisco, and that I talked about, race Mesoamerica also. Now the determinant types are these filled circles, and you see that the trait is primarily a Andean trait. You see almost no determinant in Mesoamerica. Actually, there's one example here, but otherwise it's primarily an Andean trait, and people that confirms what we had seen, what people had seen before. You could also look at, as part of that, the study, if we wanted to know where is this determinacy found, in what type of genetic background, you could look here at the distribution of wild beans, wild common bean, in the Americas, and you see it's going all the way from Mexico, northern Mexico, to northwestern Argentina, and the two groups before. So we confirmed that. And this is a fairly difficult graph to look at, and I apologize for that, but it's the gene. What you see here, PVTFL1, that's the gene itself. And the structure, it has 4 exons, 1, 2, 3, 4. And then there is another gene that was sequenced that is further downstream here. Okay. So it looked at the polymorphism for that sequence. And we were particularly interested in looking at the polymorphism that causes a loss of function and so a determinacy. And actually, you see here the major structural changes. You have the most frequent one is actually the insertion of a retrotransposon in the 4th exon. Then, so this is this. You have also a full deletion of the entire gene. So the rest of the gene here is gone. And then you have a number of other non-synonymous mutations. For example, you have indels, and so on. Actually. Now, all of these mutations are found exclusively in determinate types. None of the indeterminates have any changes beyond synonymous substitutions, which is further evidence that really this gene is the causal gene controlling determinacy. What is of interest to breeders, bean breeders, are the seed types associated with all these haplotypes. And you'll see here, this is a haplotype tree where you have the wild types are in the brown color here, and they are at the center of the tree. You have the indeterminate domesticated type, which are very often together with the wild type. And then the determinate and domesticated type are at the end of the branches of the tree. So clearly this is a derived status. The most frequent branch is this one here, the blue branch, which has the retrotransposon insertion. But you have other possible determinate mutations in the green one here. This is the same thing on the Mesoamerican side. And again, you have here, you have some mutations in the Mesoamerican group, but they are usually much less frequent. So the most important thing here is that these are the types of mutations that we found at that PVTFL1Y locus. So you have frame shift, you have a deletion of an entire coding region, you have exon, intron, splicing failures, non-synonymous substitution, and the most important one, which accounts for about 70% of the determinate types, is that retrotransposon insertion, which all of these mutations actually render the gene non-functional, and hence they develop that determinate phenotype. What is of interest here for our discussion, I've told you, why do we look at this? Well, we want to see what is the active role of farmers, and it's clear that they must have had a role. It's not just a single mutation here, the most important one, but then you have others here. These are all Andean mutation in South America, and the yellow one is actually a single Mesoamerican mutation. Okay. So clearly, the farmers do select, and in different regions they may even select the same trait. You combine these data within common bean with the data from the overall domestication in the whole genus, and in the genus Phaseolus we have at least 5 domesticated species. I've mentioned common bean. You also have here runner bean. Then you have this year bean, which is probably the least known of all the domesticated species, and it's actually on an evolutionary timescale. It's a stabilized hybrid of a cross between common bean and runner bean. So its cytoplasmic genome is more like common bean. Its nuclear genome is a mosaic of common bean and runner bean. You also have tepary bean, and then you have lima bean. Now 2 of these species, and maybe a third one, have been domesticated at least twice. So you can have anywhere between 7 or 8 domestications. So we see that the farmers not only have looked at variation within the species, but also looked at the entire genus as a potential domesticate. And why so many domestication? Well, that's somewhat of a question, but I would argue here that each of those species, these 5 species, has a different adaptation. Okay, so you have mesic, which is intermediate in terms of temperature, humidity. Runner bean is cool and humid, as I discussed before. And then year bean, which is a hybrid between common bean and runner bean, is somewhat intermediate. Temporary bean is hot and dry from the US Southwest and the Northwest of Mexico. And lima bean is probably the species with the broadest adaptation, and it's primarily adapted to hot and humid, but you can find it at higher altitude, colder and drier as well. The reproduction system is very diverse. Runner bean, for example, is almost obligatorily outcrossing by presence of bumblebees and hummingbirds, whereas temporary bean, for example, at the other extreme, is cleistogamous. Okay, so probably farmers looked at all these what we call different species, and they looked at them instead of adapting a single species to a broad range of environments. They actually conducted multiple domestications depending on the adaptation of these specific species. And this gives you a little bit of distribution of these 5 species. You have temporary bean here in yellow. This is the northwest of Mexico, southwest of the US. You have runner bean, it's primarily the southern half of Mexico and Guatemala, and the same thing for runner bean. And then the 2 species with the broadest adaptation are common bean in blue, and then lima bean in green. And you see that they go pretty much from Mexico all the way to Argentina, both species. And both species also have been domesticated twice. I think that there are a few other examples where you have multiple species that were domesticated in the genus. Chili peppers is one of them. Cotton, maybe wheat also. The hypothesis I would present is that Phaseolus is actually a fairly young genus based on the data of Alfonso Delgado. These are molecular systematics data. It's only about 4 to 5 million years old. They appeared after the last major geological event in Mexico, which is the formation of this volcanic axis
I enjoyed it quite well.
that crosses Mexico from west to east and contains these big volcanoes like Popocatépetl and Orizaba and so on. And that kind of, after that, the whole scene, geological scene, quiets down. And I think that was then the opportunity for plants in general to start spreading and speciating. And that's the same for Phaseolus. And so you have Hello, I cannot hear you.
With microphones. I see that Jamie has typed in a comment in the chat box. People interested in sustainability often complain about lack of diversity in crops and want more. Is my microphone not working?
I hear you now. Can you hear me?
It looks like we have several questions in the chat box.
Hello? Yes, I see those. So there is a first question on maximizing participatory breeding. I think clearly that we have to move towards that. That is participatory breeding can change from one area. I think that I'm also in charge of the breeding program here in California, and I'm doing participatory breeding with the California farmers, but it takes on different ways. I go to their meetings, they come to field days, and they define the priorities for breeding. So in that sense, this is certainly a form of participatory breeding. In countries like Mexico, for example, I think that in order to maximize it, I think that there are two things that may increase. I think that we have to provide additional genetic diversity that may be relevant in terms of maybe segregating populations or a wider array of advanced lines so that they can do either varietal selection or segregating selection. The other thing is to see how do you do the selection. In corn, for example, there is the example of what is called Gardner's grid, where you divide up the field in a grid, and then for each cell of the grid, you pick the best material. So to come back, it's twofold. I think there's the selection methods, and then the materials on which to act on the selection. And so they introduced themselves already in diversity. I showed you the example of the small black beans, of this Anita cultivar, but we can increase that, the influx of diversity. So did this answer my question? Okay, so the second question, what type of factor is the predominant factor for differentiation of gene pools? I think that it's a purely geographic differentiation. And it's actually more than that. It's a form of early speciation. So there are some F1 lethals that appear controlled by two complementary genes, one Mesoamerican, one Andean. And when you cross them, the F1 is lethal, but it only affects about maybe 10–20% of the crosses. So the other question is, I think that if you see two groups of cultivars, it's because it corresponds to different adaptations or different uses. For example, in Africa, we see that the seed types is also actually an indirect indicator of what type of dish or what type of conditions these beans allaying risk. It's a risk control, because if you have adverse conditions, they may not affect the different groups in the same way. And typically, you see that also in beans, for example, in Guatemala, they will grow common bean and runner bean, and they prefer common bean, but sometimes the disease pressure is so high that only common bean, only runner bean is going to survive. So the question here of John is if we have studied LD um, in wild and domesticated types. We don't have any kind of LD studies with a high density of markers. The only studies we have is a fairly low density of markers. I think that there's work underway in Mexico, also in the US, and I have a proposal to study wild beans in much more detail to get at that. What is the linkage to equilibrium in wild beans compared to domesticated beans? So it's a very good question. What I think about the cross between vulgaris and coccineus is that we need to know in these interspecific crosses, how can we transmit genes from the other species like coccineus? There are some regions which we know we cannot introgress, so we need to know that. Is Bayesian structure analysis a reliable method to analyze the structure of crop diversity? Are you talking about, well, I think it is, I would say. And in beans, you have to realize that um, we had prior results before running structure. And what structure has done is just confirm what we knew before. And it does actually provide us with some additional information, which is the composition of a genome, whether it's predominantly, you could say, in between quotes, pure, or if there is a mixture of different genomes. So in terms of kind of the result of gene flow. And in that sense, I think that it has been, for us, it has been a pretty good method. It confirms what we knew before taxonomically or prior molecular studies. I'm not sure I understand your question. Is there a minimum error of probability? Well, you talk about error combining multiple loci that are similar. In a sense, you have that in beans in terms of the separation by Andean versus Mesoamerican. We had seen this very early on, I would say 20 years ago, when we ran isozymes. There we had very few loci, and there is a very strong separation between Andean and Mesoamerican groups, and to the point where if you want to do linkage disequilibrium linkage disequilibrium studies, you have to do it separately in the Andean and the Mesoamerican group. Otherwise, you just have too many errors. But within the Mesoamerican and within the Andean, actually, the linkage disequilibrium low is decreased considerably. The question is about farming might inherently increase diversity within self-pollinated subpopulation. I thought it was just the opposite, is that it's more of a challenge to increase diversity in self-pollinated populations compared to cross-pollinated. If you take cross-pollinated population like maize, for example, there is already a tremendous amount of diversity. It's in the self-pollinated subpopulations. There, the researchers would have to provide segregating populations, which you don't have to do with cross-pollinated populations. But maybe I misunderstood the question. Yeah, I think that there is definitely to increase diversity, but what we see, the example I gave you, is that
thanks again, Dr. Gepts,
some farmers already do this. They are able to increase diversity to a level that is comparable with the wild population surrounding the field.
Sticking out for the end of the webinar as well. Our next TCAP webinar will be in 2 weeks on April 17th.